Hello Year 9. So this video is for the answers to last week's homework. So you need to have the homework out in front of you. If you haven't done the homework, you need to quickly pause the video and do the homework before you watch this. And you should have everything else out ready because you've already done yourself a 10-question quiz. If you haven't done a 10-question quiz, maybe pause the video now and do it before carrying on. And you've got all your distractions out of the way. So you know your phone's not going to be beeping and distracting you, and not listening to music or anything like that, so you can hear me properly. So, explanation of why all the answers are written on here. So I just made a video and it didn't record properly, which is why all the answers are written down. I don't have another copy, so what I'm going to do is I'm just going to talk through the answers, but you need to still listen carefully, because there's a few conversion questions which you need to think carefully about, and I'll go through those on another piece of paper. So, what is the typical size of a plant cell? 10 micrometers to 100 micrometers, because they're eukaryotic cells. How many micrometers are there in 1 meter? All right, so for that one, I'll just give you a quick run through of how that works. So 1 meter equals 1,000 millimeters, and that's something you should remember. Okay, you might remember it by thinking, well, I know that 1 centimeter has 10 millimeters and a meter has 100 centimeters, and 10 times 100 is 1,000, but really it's best if you just remember 1 meter is 1,000 millimeters. And also you should know that 1 millimeter is 1,000 micrometers. Okay, so if there's 1,000 micrometers in a millimeter and there's 1,000 millimeters in a meter, how many micrometers in a meter? Well, it's obviously just going to be 1,000 times 1,000. So that's how I get 1,000,000 micrometers. All right. Why do microbiologists stain cells before looking at them with a microscope? To see them better. All right. So you know, if you've given them more detail than that, fantastic. You know, if you talked about staining it with particular dyes to see particular structures, that's great. In general, which are bigger, eukaryotic or prokaryotic cells? Eukaryotic. Where is the genetic material stored? In a prokaryotic cell, that's in the cytoplasm. Remember, in a eukaryotic cell, it's in the nucleus. That's one of the key differences between eukaryotic and prokaryotic. I hope, by the way, that you've all been practicing saying eukaryotic and prokaryotic, you know, telling everyone at home all about the interesting cells you've been learning about. Animal cells are a type of what kind of cell? That's eukaryotic. Would you expect a human cheek cell to have a cell wall? No. A cell wall is a subcellular structure found in a plant cell. So a human cheek cell is not a plant cell, it's an animal cell, so you'd expect it not to have that. Mammalian sperm cells have a flagellum. Okay, what is a flagellum? So a flagellum is a subcellular adaptation to help the cell swim. All right, so just to remind us, this word subcellular, so sub means smaller than or underneath. So when I talk about subcellular, I mean a part of a cell. So you know that a cell, you know, might look like this. That's the nucleus, and you've got these mitochondria. So that's the whole cell, whereas the nucleus, that's one subcellular structure. Okay, so the flagellum, you'll remember, are these tail-like things that come off the cell, and they help them swim. Suggests why mammalian sperm cells possess this adaptation. So for this, you need to go right back to when you learned about reproduction, and you'll remember that the sperm fertilizes the egg, but in order to do that, the sperm needs to get to the egg, and the flagellum helps it move through the body towards where the egg is, so it can fertilize it. Why don't human cells contain chloroplasts? That's because they don't do photosynthesis. Remember, chloroplasts is where photosynthesis happens, so if you don't do photosynthesis, you wouldn't have chloroplasts. Respiration occurs in mitochondria. The chemical equation for this is this. Identify the reactants of the products. Reactants on the left-hand side, products on the right-hand side. How many carbon atoms are in a molecule of glucose? Glucose is this thing here, C6H12O6. The little 6 after the carbon means 6 carbon atoms. What is the law of conservation of mass? It means mass cannot be created or destroyed. So you've got 6 carbon atoms over here, which means that over here you need to have 6 carbon atoms as well. You can't get more carbon atoms, or you can't lose carbon atoms. Whatever you start with, you have to end with. But we'll do more about that in the next lesson. Why do cells do respiration? That's to produce energy, of course. Copy and complete the sentence. Lungs contain many alveoli where oxygen passes into the bloodstream by diffusion and carbon dioxide passes out of the bloodstream by diffusion in a process called gas exchange. I'm very confident that you all got that one right. Some of you may have thought that this here was gas exchange, but you remember that the movement between the alveoli and the blood, that's diffusion, and the overall process is gas exchange. Okay. A pupil is, so this is number 11, a pupil is looking at some human cheek cells through a microscope. She wants to see a bigger image than the one she currently... Oh no, sorry. What am I doing? Number 11. A pupil is looking at some onion cells through a microscope, but can't see anything. What would you suggest he tries doing first? Okay, turning the light on. So you'll remember in the microscope, okay, there's a light source at the bottom, because otherwise, you know, you look in here. All right, now if there's no light shining up, you're just looking into a dark tube. All right, so there's no light going into your eye. If there's no light going into your eye, you won't see anything. That's what the dark is. All right, so you need to have light shining up through there. So if it's dark when you look into your microscope, so try turning the light on. A pupil is looking at some human cheek cells through a microscope. She wants to see a bigger image than the one she currently has. What does she need to do? She needs to change the objective lens. You change this lens here by rotating that part of the microscope. Okay, and you might have used it already, but you'll see it next year. You don't make the image bigger by twisting these knobs. Okay, that just makes the image go in and out of focus. If you want to make the image bigger, you have to change the lens. All right. 13. What is the first thing you should do when preparing to look at a slide through a microscope? That is, you need to move the stage to its lowest position using the coarse focus knob. 14. A pupil is looking at an animal cell through a microscope. The eyepiece lens has magnification 10 times and the objective lens has magnification 100 times. What is the total magnification? Well, if you've got 10 times and 100 times and you add those together, then you're going to multiply your image by 10 times 100, which is 1,000. The animal cell has an actual length of 12 micrometers. What length does the image of the cell have? So, if the actual length is 12 micrometers, and I've got 1,000 times magnification, it's going to be 12 micrometers times 1,000, which equals 12,000 micrometers. Now, this isn't really very helpful. It's difficult to imagine how big 12,000 micrometers is. But you'll remember, of course, that 1 millimeter equals 1,000 micrometers. Okay, so we have here 12 millimeters. All right, and to make it easier, you don't need to do this bit, but just so you understand, that's 1.2 centimeters. So we've gone from this thing that's so small you can't see it, but we do a huge, huge magnification on it, and now it's quite large. All right. 15. A pupil is looking at a plant cell through a microscope. The eyepiece lens has magnification 10 times, objective is 40 times, so it's 10 times 40 is 400. The plant cell has an actual length of 15 micrometers. What length does the image have? Okay, so it's going to be 15 times 40, which I'll need my calculator for. So that's 15 times 40 is 600 micrometers. That's not right. What have I done? Oh, it's 400, sorry. So it's 15 times 400, so it's 6,000 micrometers, which again is 6 millimeters. Okay, all right, and the last question. Okay, so a red blood cell has a diameter of about 10 micrometers. How many red blood cells would you need to put side by side to make a line 1 centimeter long? All right, so let's have a think about this. All right, let's imagine we've got a line which is 1 centimeter. Now I know that's not how big a centimeter really is. Okay, I do know that. You don't need to email me and tell me that's not how big a centimeter is. Okay, that's how big a centimeter is. All right. But what I'm doing is, to make this easier to understand, we're going to pretend that we've sort of, maybe, if you want, you can think about it, we've zoomed in a little bit so we can see it's bigger. All right, but it's still a centimetre. Now, I want to know how many 10 micrometers I can get in my centimetre. All right, so what I need to do is I need to change my 10 micrometers, change my, I'm not going to change the micrometers actually, and what I'm going to do is I'm going to change my, because they need to be in the same unit, so this unit and this unit need to be the same. So I'll change my centimetre, that'll be easier. All right, so I want to change 1 centimetre into how many micrometers? Okay, well, I know that 1 centimetre equals 10 millimeters, and I know that 1 millimeter equals 1,000 micrometers. So that means that 1 centimeter equals this number times this number, because if I've got 1,000 micrometers in a millimeter and 10 millimeters in a centimeter, then I'll have 10,000 micrometers in a centimeter. So I'm going to rewrite that as, it is still 1 centimeter, but it's also 10,000 micrometers. Now, if I've got a red blood cell, or my red blood cell here, this is 10 micrometers, from one side to the other side. How many of those can I fit into 10,000 micrometers? Well, that's an easy sum to do. It's just 10,000 divided by 10, which is 1,000 red blood cells. Okay, so good job if you got that one. That was a little bit of a tricky unit conversion. Now, next, what we have is how many red blood cells would fit into an area of 1 centimeter squared. So I know that if I have, let's draw a square out, okay, and the square is 1 centimeter by 1 centimeter. I've already worked out that in this 1 centimeter here, I've got 1,000 red blood cells, okay? And if along here, that's 1 centimeter as well, then that's also going to be 1,000 red blood cells, which means that in this area here, I'm going to have 1,000 times 1,000 red blood cells, which is 1 million red blood cells. And finally, in a volume of 1 centimeter cubed, well, if I have a cube, all I'm doing is I add another length onto it, and now the number of red blood cells inside this cube is going to be the number along here times by the number along here times by the number along here, which is 1,000 times 1,000 times 1,000, which is 1 billion red blood cells. And I'm not sure what the exact number is, it might vary depending on other things, but it's a good estimate of how many red blood cells would be in a centimeter cube, because that's how big a red blood cell actually is. All right, so you think about how a centimeter cube is quite small, and you've got 1 billion red blood cells in there. All right. Anyway, so well done on that. If it was too quick at any point, pause it, go through it again. Otherwise, you can now move on to the new material for this week.